Today's lesson is going to be on genetic diversity and adaptation. So we're in chapter 9 and that's section 9.3 of chapter 9. Your starter today is looking back at meiosis. So could you please describe and explain the processes that occur during meiosis that increase genetic variation? This is a 5-mark question, so make sure you've got 5 good marks down in order to get the full 5 marks. So pen and paper ready, please. And could you pause the video now while you do this? Right, so I'm going to assume that you have completed that. And let's talk about, sorry, 1 second, going the wrong way. Right, so let's talk about how genetic variation is, sorry, I'm just trying to get a little spotlight or something up here. Okay, that should do it, right. No, I haven't got it. All right, one more try. Sorry, I'm just fiddling around with it, trying to figure out how these things work. So there we go. Okay, so I'll just use this for now. So trying to describe genetic variation in meiosis. So if we look at this picture over here, you can see that there are 2 possibilities if you have a cell that has got 2 pairs of chromosomes. So we can see that this cell has got 2 pairs of chromosomes. So that's 1 pair of homologous chromosomes, and that's the other pair of homologous chromosomes. And here it is again, and here it is again. So they're the same cells, right? They're just looking at 2 different possibilities. From the worksheet that we did recently, we know that we can calculate the number of possible combinations if we know how many pairs of chromosomes there are. So there are 2 pairs of chromosomes. There are 2 pairs of chromosomes. 2 to the power of 2 is 4. So there are 4 possible combinations. If we look at the bottom, we can see there's 1 combination, 2, 3, 4 possible combinations. How do these different combinations arise? They arise through the independent segregation of the homologous chromosomes. So the homologous chromosomes separating differently to one another, going into different cells in different combinations in meiosis 1, okay, in metaphase 1. So in possibility 1, what we find is that the blue version of the chromosome from each homologous pair goes into one cell, and the red one goes into another cell. That means that these are the possible combinations of the gametes after meiosis 2, after metaphase 2. So you have this combination and this combination arising from the possibility 1. Possibility 2, in metaphase, the homologous chromosomes of different colors go into different cells to one another. So you end up with these possible combinations when the gametes are made. Okay, so it's this the homologous chromosomes separating from one another, how they separate from one another in meiosis 1. So at this stage over here, okay, meiosis 1, that is where independent segregation of homologous chromosomes happens and has the impact. Okay. The other way that genetic variation is brought in meiosis is through crossing over. So if this is our homologous chromosomes, if these are our homologous chromosomes, so remember, homologous chromosomes are a chromosome pair. One of them is maternal, one of them is paternal. Here they are replicated. We know that they are replicated because each chromosome, so that's 1 chromosome, each chromosome is made of a chromatid. 1 chromatid, sorry, it's made of 2 chromatids, 2 chromatids. So 2 chromatids make 1 chromosome, okay? Those homologous chromosomes, when they align, then they will cross over with one another. And what that means when they cross over over here is they swap bits of the chromosome with one another. So part of this chromatid will swap with part of this chromatid. So this is what you end up with. So this chromatid over here is as it was in the beginning. It's not recombined. This chromatid is now recombined. So they started off identical because obviously they were identical copies of one another. Okay, they are now non-identical. Okay, non-identical. One of them is recombined. Same here. This one's recombined and this one isn't. Okay, so this is also introducing a degree of variation. So in your mark scheme, this just turns out to be the mark scheme for that particular question. The things that you should be talking about are independent segregation of the homologous chromosomes. So if you said independent segregation of homologous chromosomes, that would probably take in all three of those marks. Crossing over, and these create new combinations of alleles. So actually explaining what the effects of those are. You can pause there if you want to copy that down. Okay, so today we're talking about genetic diversity. What is genetic diversity? You should hopefully have some sense of what it is. Genetic, genes, diversity, difference. So it's the difference in our genes. But specifically, we're talking about the genetic diversity in a population of individuals. So genetic diversity is the number of different alleles of genes in a population. So if you remember, when we talk about genes, we can talk about genes for example, flower color. So a sweet pea might be red, or it might be white, for example. So there is a red, a gene for flower color. The alleles, however, will be a red allele and a white allele. So we're talking about the alleles, we're talking about all of the possibilities of how the genes can be expressed. Okay, so if we have a look at this photograph of the corn, that's a nice example of genetic diversity, showing lots of different colors of corn kernels, and those will be down to the different alleles for the color of corn. And so you can see there are alleles for yellow, for white, for black, for brown. There are alleles for blue as well. There is a famous Mexican blue corn, and they make blue taco chips from it. Dogs. We all know that there's great amount of genetic diversity in dogs. We all know that a Chihuahua and a Great Dane are in the same species, but clearly they're massively different. So there's great genetic diversity in species of dogs. Lots of combinations of alleles there. The finches that Darwin came across in the Galapagos Islands when he came up with his ideas of natural selection, all of the different beak types are described. Those phenotypes are described by the genotype, and the genotype is combinations of different alleles. So all of the alleles for all of these different beak types would have been present in the original population of finches that went on to populate all of the different islands in the Galapagos Islands. So by these birds having these different alleles, they were able to adapt to the changing conditions, to the new conditions on the different islands, so that they could maybe break heavy seeds more easily, or they could pick up small berries more easily, or whatever it might be. But these alleles needed to be there. They needed to be in the gene pool in order for the animals to in order for them to be able to evolve in this way. Okay. When we look at a population, for example, this population of flamingos, they all look exactly the same. So phenotypically, when they all seem identical, but there will be massive genetic variation in there as well. We're not aware of the genetic diversity in a population until we actually either look at the genome itself to see the differences in the genome, or we see the population being challenged by some change in environment, and that change in environment then exposes the fact that there are different alleles present. I just put this picture in because they're really pretty butterflies, but it's a very nice representation of genetic diversity, particularly at this time of year. It's spring, sun's out. If you're sitting in the garden, you've probably seen a fair few butterflies, but these are most likely different species, but it's just showing that you get, obviously, genetic diversity within a population, but genetic diversity between different species, so genetic diversity within a family of living organisms as well. So genetic diversity, what do we need to know? We need to know that genetic diversity is the total number of different alleles in a population. So the more, the higher the number of different alleles in a population, the higher the genetic diversity. Okay, what is a population? A population is a group of individuals of the same species that live in the same place and can interbreed. So you might have a group of deer on one side of a mountain and a group of deer on another side of the mountain. They are the same species by all accounts, but they live in different geographical areas. So they are two separate populations. Okay, and they're two different separate populations, and they will be subject to different environmental pressures. So would potentially be very slowly evolving differently, but they can be the same species, just different populations. The greater the genetic diversity, the greater the chance of surviving any environmental change. Without genetic diversity, there is no opportunity for natural selection. You have to have genetic diversity in order to be able to evolve via natural selection processes. There have to be a variety of alleles present that can possibly respond to those changes in environment. So let's talk about natural selection now. That's something that's familiar to you from GCSE. So if you have a look at this picture, it should be a story that is known to you. You would have learnt about this at GCSE. So if you pause the video now and just remind yourself of what is actually going on in this picture. Tell yourself the story of natural selection using this picture. Right, so hopefully you've revised these ideas. So I'll very briefly just discuss them again. You should remember that this is a story of the peppered moth, and the reason why it's such a well-known and well-described example of natural selection is because it's natural selection in action. We can actually see it, observe it happening. And scientists did observe it happening, and they documented it very, very well. Originally, peppered moths were light coloured so that they could be nicely camouflaged against the light coloured trees. Within the population of peppered moths, there were a variety of alleles for wing colour. There was the lighter colour, there was maybe a sort of a greyish colour, and then there were darker colours, darker grey, and possibly even black. So there was a range of alleles, so a range of phenotypes. Most of the moths were light coloured. The reason was that the darker moths were far more visible on the lighter bark, so the birds would eat the darker moths. Because there were more white moths available, the white moths would be more likely to reproduce with other white moths. So the allele for the white-winged moth would be passed down to offspring, so that over generations, most of the moths would have the white-winged allele. But of course, these black-winged alleles were still present in the population. Remembering, you have two alleles, so you will be able to carry alleles for different colours. When the Industrial Revolution came along, as you know, lots of soot was emitted into the atmosphere. That soot deposited on buildings and it deposited on trees. And so the trees turned from lovely white, maybe silver birchy bark to grey bark, and the white moths were now the ones that were not particularly well camouflaged against this dark backdrop. So the birds are picking the white moths off. This meant that even though there were only very few black moths to start with, because the black moths were not getting picked off, because they were not getting eaten, they were more likely to then go on and reproduce. So when they reproduced, they were passing on the black allele. So the frequency of the black allele, the black-winged moth allele, increased in the population for the offspring and the further generations. And that continued until most of the moths then appeared to be black, although of course there were still white moths around because the white allele is still present in the population. So that's natural selection. And the description is using this sort of terminology that we're expected to use now at A level. So this is a sort of a kind of stock answer that you might give when being asked a natural selection question. It's unlikely that an examiner will ever say, describe the process of natural selection, although it is possible. Usually they'll give a context, they'll give you an organism, and they will ask you to describe how that particular organism might have evolved. So the first thing that you need to acknowledge is that there is genetic variation within a population, okay? And that genetic variation within a population we refer to as the gene pool. So the gene pool has a large variety of alleles. So that's saying there is a lot of genetic variation. Where does this genetic variation come from? Well, it would have always come from, at some point, a random mutation. So any differences come about because a mutation event has happened. Those mutations may be advantageous, they may be disadvantageous, they may be completely neutral. Mutations are happening all the time, and more often than not, we're not aware of the fact that we have a mutation until the environment challenges us, and then that mutation comes to the fore. So a nice example is the very few individuals in the world who are resistant to HIV. They're resistant to HIV because they do not have the receptor molecules on their lymphocytes for the virus to bind to. They would not know that, but they would not know that at all unless they got challenged with HIV, unless they get HIV and survive it. So most of them are unknown to us. Okay, so we have a lot of genetic variation. That variation comes from random mutations, which creates new alleles. These alleles may confer a competitive advantage over the rest of the population. So possibly the black allele, the black-winged allele for the peppered moth, gave it a competitive advantage over the rest of the population. They were then more likely to survive, then they reproduce, and they produce more offspring, okay? So then the offspring had this new allele because the new allele has been passed to the offspring, and then over generations, the number of individuals with this allele starts to increase at the expense of the less advantageous allele. And this is the key term, the frequency of the new allele increases. So we always need to be talking about allele frequency increasing in the gene pool. Okay, so that's another key term that we want to be using in a good answer. All right, very simplistic drawing showing some evolution. If that's a little organism, it reproduces, and there's another little organism. So sometimes the little organism reproduces, there is a mutation, a random mutation, and it results in a different phenotype. In this particular example, the little organism loses its little spikes. Clearly it's an aquatic organism, and so there are predators out there. The predators will then try their best to get them. Or the spiky one, he can't be eaten, clearly, because he's spiky, whereas this one, well, he's just gobbled up straight away. So that means he has no opportunity to pass his alleles down. He has no opportunity to reproduce. He's no opportunity to reproduce, pass his allele down into his offspring, and then down into further generations. So this allele that made this particular phenotype is lost from the gene pool. Okay. Had it have been an advantageous one, then it would be kept in the gene pool because this would be able to go on and reproduce, and they would and would thereby increase the allele frequency for that particular allele. Okay, so what do I want you to do with your with this information? So there's a couple of things. The first one is to click on the Cricut link. So here it is, over here, and read the comic and answer the questions. And then click on the PhET link underneath it and play around with natural selection. Observe how the allele frequency change with different environmental pressures. For the PhET link, some of you might come up against some problems depending on what operating systems you have. It is a lot of fun, so if you can change the permissions on your computer so you can use the PhET, please do it, because there are other animations in there that you will find useful in your biology, but if you do other sciences as well. Make some summary notes of the whole of 9.3, answer the questions on page 230, answer the Seneca questions that I've set on Seneca, log into the Khan Academy, oopsie daisy, and do the evolution assignment. So that's going to be watching a video on evidence for evolution and complete the practice questions. And then there's a couple of exam questions that I'm going to give you. So could you complete those as well? Okay, I'll put this PowerPoint on the assignments tab so you can have a look at it. It's got a little bit more information about natural selection on it as well. Okay, so the other things that I wanted to show you here, let's just do that. Okay, let's just close all of, okay, so this is your specification. So just please keep looking at your specification. It's really, really useful. So we've just completed genetic diversity can arise as a result of mutation or during meiosis. So meiosis and mutation increase genetic variation. So here's your description of meiosis. Be familiar with this. You've just been doing these calculations over here, using expressions, use the expression 2 to the power of 10 to calculate possible numbers of different combinations of chromosomes. So you've been doing that. And now we're moving on to this, genetic diversity and adaptation. Okay, so principles of natural selection, random mutations, advantageous alleles inherited, blah, blah, blah. So next lesson, we'll be talking about directional selection, stabilizing selection. Okay. So that's just a recommendation. Really keep your eye on your specification. It is a very, very useful resource. And here is the PhET page. You should go to this one over here, natural selection. And here it is. So it's a little animation and you click on that and you can play around with it. Your textbook. So these are the summary questions I want you to do. Seneca learning will be probably on this genetic diversity, natural selection. Khan Academy will be this, evidence for evolution. You'll be watching this video and then there's some questions afterwards. And then I have put on the TEEB in the post area. This is just for your interest if you want to. There is the deep evolutionary history of the new coronavirus, because this comes from the Understanding Evolution website that I've shown you before, and it gives you a nice little sort of evolutionary tree showing you where COVID-19 and other coronaviruses arise, shows the links between COVID-19 and other coronaviruses that cause respiratory illnesses. And yeah, that's it. So it's not a lot, but it's just quite interesting. It's really nice biology. Okay, so good luck with that. And I will be setting this on Thursday.